One of the most compelling aspects of early embryonic development is the remarkable series of cell and tissue rearrangements that gives rise to the diversity of anatomical forms that we commonly associate with all animals. As a consequence of these movements, embryos generate mechanical forces that act both locally and over long distances to influence not only morphogenesis, but also cell signaling and cell fate decisions. Thus, understanding how cells generate, propagate, and respond to physical forces is a key question. The cell and tissue movements of gastrulation in the amphibian xenopus have been described in considerable detail over the past several decades. In the mesenoderm, a collective form of cell migration is observed, whereby a contiguous group of cells advances directionally using an extracellular matrix substrate assembled along the inner surface of the blastocoel roof. Each cell within the mesenodermal mass extends protrusions in a monopolar fashion that are directed toward the animal pole of the embryo. Unlike the situation for single cells, in the mesenoderm, leading-edge traction stresses generated at the ECM substrate are balanced at the rear of each cell by intracellular stresses located at sites of cell-cell adhesion, depicted here in red. It has been known for some time that monopolar directed motility in this system and other collectively migrating tissues requires that the cells remain adherent to one another. However, when dissociated to single cells and plated on fibronectin, single mesenodermal cells become multipolar protrusive and migrate in random directions. While chemotactic and other mechanisms have been suggested to control the directed migration of intact mesenoderm, the role of mechanical force in regulating individual cell behaviors within the collective had not been addressed. We devised an experimental strategy that would enable us to ask whether local forces applied to single mesenodermal cells in isolation are sufficient to induce monopolar protrusive behaviors and directed migration.
We felt that in order to understand the emergent properties of a collectively migrating tissue, it would be most informative to replicate in single cells the behaviors displayed by cells when part of a group. A magnetic bead coated with the extracellular domain of C-cadherin was added to a multipolar mesenoderm cell plated on fibronectin. In essence, the bead mimics a neighboring cell's adhesive interactions. We used a magnetic tweezer to pull on the bead and apply tension to the cadherin adhesion, similar to how neighboring cells apply stress to one another in the tissue. When the bead is pulled using the magnetic tweezer, the cell exhibits monopolar protrusive behavior and migrates away from the direction of applied force.
These initial experiments clearly indicated an important role for C-cadherin in this dramatic mechanoresponsive behavior. Because C-cadherin is a classical cadherin, we first focused on the involvement of the actomyosin cytoskeleton in this process. Although actomyosin contractility is undoubtedly important in cell migration, it soon became apparent that intermediate filament cytoskeleton associations with these cadherin adhesions were key players in this force-dependent mechanism.
Keratin intermediate filaments, shown here in green, are strikingly localized to the basal posterior of polarized migratory cells, and are recruited to cadherin adhesions in a force-dependent manner. Moreover, the catenin protein plakoglobin forms discrete punctae that nicely co-localize with keratin filaments at cell-cell contacts. Through morpholino knockdown of plakoglobin and keratin, we were able to show that both these proteins are crucial for the polarization of cells in response to force application on cadherin adhesions.
We don't yet have a complete molecular picture of these mechanosensitive cadherin adhesions, but ongoing studies in our group are aimed at finding other proteins associated with this complex. We are especially interested in identifying proteins undergoing conformational changes in response to force, suggesting a direct involvement in the transmission of mechanical signals.
There are many exciting questions that this research opens up. For example, it remains a mystery exactly how plakoglobin and keratin are involved in directing cellular protrusive behavior. Are keratin filaments actually bearing load when tension is exerted on cadherin adhesions? And of what consequence might this be to the cellular response? In light of our findings, we now need to examine the role of the actomyosin cytoskeleton in this system. The interactions between these different cytoskeleton networks are likely to play a central role in the cellular response to mechanical stimulation of adhesions.
We suggest that forces acting on cell-cell contacts have a fundamental role in guiding protrusive and migratory behaviors. We have termed this phenomenon cohesotaxis to reflect the importance of cell cohesion in directing the individual behaviors of cells within a collectively migrating tissue. It will be necessary in future to establish how cohesotaxis acts in concert with other migratory guidance mechanisms, such as chemotaxis and durotaxis, to promote collective migration in multiple physiological contexts, from embryonic development to wound healing and the progression of tumor metastasis.